Okay, good afternoon, everyone, and welcome to the front row. My name's Jamie Williamson. I'll be your host today at the front row. This is, we took the summer off. This is the first of 4 lectures in the second half of our season. And this is the 8th season of Front Row. So we have, looks like about 200 people here in person. So it's good turnout. And it seems like we have about 5,000 people online. So yeah, this is good. So for those of you in the virtual audience, we encourage you to come to campus. There's a few seats left, not many, but come to campus, and it's a good experience. So, but welcome to everybody. So today's topic is cancer. There's nobody in this room that hasn't been affected either personally or in immediate friends and family by cancer. And there are many therapies: chemotherapy, radiation, and immunotherapy, which is the subject of today. And the disease is serious, and the treatments are very serious. So one of the major problems with cancer treatment is that it's very toxic, and so that's there's no exception for the case of immunotherapy. So the thing we're going to hear about today is CAR-T cells. So T cells are the natural cells in your body that attack foreign antigens, and that's been used as a treatment for cancer. So you can take cells out of a patient, program them to attack a certain kind of cancer cell, and then they go. And one of the drawbacks with this whole therapy is that it can be very toxic. The cells can just go haywire and start doing things they're not supposed to, the T cells, or once they're done eating the tumor, they go on to their second favorite thing, which could be some essential body part. So there's very serious drawbacks, even though it's a very promising therapy. And so what Travis has done is engineered out some of these shortcomings in the T cell therapy. And this has been done in the context of the Calibr-Skaggs Institute. So we have 100 faculty doing basic research on campus, and Calibr is the translational arm where they're using not-for-profit setting to develop next generation medicines. And so the T cell program that Travis is going to tell about is one of the leading programs. It's very promising. It's going to be exciting, and I'm sure you're going to be impressed by this. So we're a not-for-profit. Calibr's one part. We have basic research, and we have a graduate program. And those are really the 3 legs on the stool of our success. We have a tremendous faculty. We have tremendous translational scientists. Many of you heard the last lecture from Eric Topol. So we've got personalized genomic medicine, and it's really a remarkable place. And one of the things we really do is train the next generation of scientists. So this is all done by funding. Our primary source of funding is the federal government. Yeah. It seemed, I think I was really whining about it the last couple of front rows in the spring. It looks like Congress has at least mitigated some of the major impacts of the proposed budget. And we're a little bit more optimistic, but nevertheless, we're under some serious funding stress from legislation. So if you feel so inclined, we would appreciate it if you'd raise your hand and voice to your representative or senator to stress the importance of research. It's making new discoveries that enable new medicines and training the next generation of scientists. Please. The other sources of funding are philanthropy. We've had some major gifts. The Skaggs family endowed the Skaggs Calibr Institute. We have partnerships with private industry, and many of you are individual donors sitting in this room who have helped us. And the impact of donations is that we can use them to seed programs that are not quite ready, new ideas that can't get the very competitive federal funding. So everybody can make a difference, and we encourage you to interact with the staff outside during the reception at the end. Now this is, before I introduce Travis, a couple of business points. So first of all, this is about cancer. Neither Travis nor I are MDs. We cannot recommend treatment or provide any kind of medical advice. So please, we need academic questions that I know everybody's interested, but we can't be your proxy physician for this afternoon. So the other thing that we'll do is at the end, there'll be Q&A. So if you're in the virtual audience, you can type into either chat or Q&A. That will get relayed to me. If you're in the physical audience, put your hand up. And it's hard for me to see, but our staff has a lightsaber that they'll wave, and it'll get my attention, and we'll call on you from the dark. We'll try to get to as many questions as we have. We'll come back, and Travis and I will have a little chat. So we're looking forward to that. So Travis got his PhD here at Scripps, working with our president, Pete Schultz. He did a postdoc at Harvard, and he returned to join Calibr. He's now the vice president for biologics. So I'm going to let Travis tell you a little bit more about his personal journey to get here. But for the moment, please join me in welcoming him to the stage. Travis. All right, take it away.
Thanks for the introduction, Jamie. I'm Travis Young, as Jamie said. I've been part of the Scripps Research family now for almost 20 years, having done my PhD training here. And I came back to do this, to build the group in biologics at Calibr-Skaggs and to work on cancer immunotherapies. I gave one of the first front row lectures almost 7 or 8 years ago now, and this has really grown incredibly since then. And I'm very excited to tell you about the progress we've made since the last time I was here. So if you're here, you already know that Scripps is recognized as the world leader in biomedical research, ranked #1 in innovation, 6 Nobel Prizes, a highly awarded faculty. The list goes on. And as a scientist, I need to disclose my bias. I obviously did my PhD training here, and my boss is the CEO, so I have to say this. But what is the impact of all of these accolades? What does it mean for you? And the answer is new medicines. Over 100 years of developing innovations that have led to drugs like Humira, Benlysta, Trodelvy, Zeposia, and many, many more. These drugs would not exist if it were not for the innovation and the inventions made here at Scripps. So today, I want to highlight 3 things about what we're doing as we look towards the future. The first, as Jamie mentioned, is I want to describe how Scripps is changing the model for nonprofit drug discovery research by building an internal translational research engine called the Calibr-Skaggs Institute for Innovative Medicines. The second is I'll talk about how we've used that model to develop a gene and cell therapy for cancer and for autoimmune. And then the third is how that therapy called switchable CAR-T cells can potentially be applied to leukemia and breast cancer and even autoimmune indications. So how do you take an idea that's born in the lab and translate it into a new drug? Well, you have to go through multiple phases of clinical testing and then ultimately get FDA approval to market that drug. But the challenge is you hit something in drug discovery called the valley of death. It's the stage between discovery and approval where this excitement of the new finding hits the reality of needing to demonstrate that it's safe and it's effective in people. It's the point where costs really start to escalate. The risk remains relatively high, and therefore the value of what you have is not really fully appreciated. Bridging this gap requires more than good science. You need teams that know how to navigate the regulatory landscape, that know how to manufacture a clinical grade product, that know how to run clinical trials, that know how to protect the intellectual property, and that know how to engage with partners. Now, for those who persist through this phase of drug development, they're rewarded with a huge value inflection point in that program. But most universities and institutions, as Jamie alluded to, they never see this value inflection point because they hand off the discoveries to pharma or to startups who have the expertise and the know-how and the infrastructure in order to progress these things forward to approval. And that model has been successful. It's resulted in the approval of all of the drugs that I showed you on the last slide. But handing off the discoveries means letting go. It means letting go of the science. It means letting go of the know-how. And importantly, it means letting go of the potential financial upside that could fuel more discoveries. And again, as Jamie said, that's more important now than ever with the uncertainties in federal funding. So that's why here at Scripps, we've now built the infrastructure to translate things through the valley of death in a vision of doing nonprofit, bench-to-bedside drug development. And that vision is to bridge the breakthrough findings that we have at Scripps with the expertise and the know-how and the infrastructure in order to get these to patients. And in doing so, we believe we can create a model that reinvests the revenues from those successes and amplifies the impact of philanthropic and federal funding in order to create a self-sustaining, evergreen ecosystem for doing innovative nonprofit research. This is the model that Calibr-Skaggs was founded on. It was the vision of Pete Schultz. We were established in 2012 to translate things into the clinic. After 2018, when Pete took over as CEO, we merged Calibr-Skaggs into Scripps to become the translational research arm of Scripps Research Institute. One way to think about us now is like a biotech or an incubator embedded within the Scripps research ecosystem. And we are a conduit for new ideas to reach patients, but not only for them to reach patients, but to do it in a more efficient and an accelerated time frame at a fraction of the costs of what it would typically cost to do in industry. So to do this, we've developed outstanding relationships with partners. We partnered with the Gates Foundation in infectious diseases. We've partnered with major pharmaceutical companies like AbbVie and Gilead. And these relationships are so essential to the model. We excel at the discovery and the de-risking in this valley of death. Pharma excels at ultimately getting the drug to approval and marketing. And so this is the synergy that works between the Calibr-Skaggs model and getting drugs to the market. This isn't the only way that you can do it. There may still be instances in which it makes sense to out-license a new invention rather than developing it here at Scripps. There may be strategic partners that can do things better than us. There always will be. But Calibr-Skaggs is one new important tool in the ability for Scripps to do things right here. And as Jamie mentioned, in 2023, we were fortunate enough to receive a transformational gift from the Skaggs family to add Skaggs to the Calibr name, and now we are the Calibr-Skaggs Institute for Innovative Medicines, and that's allowed us to accelerate the mission even faster. I've been fortunate enough to be part of this journey since the beginning, since 2012, in building the group in biologics and in running the clinical trials in oncology. And I can truly say that no other research institute in the world is doing it like this. In addition to what you see here, we've brought in all of those other pieces for intellectual property, for business development, for clinical regulatory, in order to bridge the valley of death. And the proof of this is beginning to be seen in the output. To date, we put 13 programs into the clinic, either ourselves independently or together with partners across a really wide range of different indications. You can see here, we work in infectious disease, as I mentioned, with the Gates Foundation, oncology, immunology, metabolic disorders, regenerative therapies, and in neuro. And we've done this across all modalities. Right now in the clinic, we have a small molecule, a biologic, a peptide, and a cell therapy all at the same time. The pace of this is actually accelerating. Of those 13 clinical trials, more than half of them started just in the last 2 years. So we're reaching this inflection point now where all of the efforts in building this infrastructure is beginning to yield a steady stream of new potential medicines that are entering the clinic. And somebody's calling for one right now. Today, I'm going to tell you a story about how we brought one of those things to the clinic, a gene and cell therapy for cancer and autoimmune. But to tell you that story, it's important to understand the origin story of immunotherapy. Cancer immunotherapy, or what's also called immuno-oncology, is the idea of repurposing or re-educating a patient's own immune system to fight cancer. It's this idea that cancer is as much of a disease of the malignant cell transforming into a tumor as it is a disease of the immune system's inability to recognize that malignant cell. This idea is not new. William B. Coley was a surgeon over 100 years ago, and he was treating patients with inoperable sarcomas, and he noticed that some of these patients had spontaneous remissions of their sarcomas after getting a streptococcal skin infection. And so he was really interested in this. He was at Memorial Hospital, which is now Memorial Sloan Kettering in New York City. And what he did was he made a mixture of heat-killed bacteria, and he began injecting that into his cancer patients. And by the turn of the century, he had treated hundreds of patients with this thing he called Coley's toxins. As you can imagine, people got really sick from injecting these toxins to them. But some people had complete remissions of their disease. Now, also, as you can imagine, this is not really well-controlled science, injecting toxins into people. And so he was largely ridiculed at the time. And in the early to mid 20th century, chemo and radiation became the standard of care. And the Coley's toxins and immunotherapies were largely forgotten. But it's interesting to think that immunotherapy actually predates chemo and radiation because we consider those now as the pillars of cancer care. But unfortunately, again, for 100 years, it was kind of forgotten. You fast forward 100 years to the 1990s, and Tatsuko Hanjo and Jim Allison, who did his postdoc training here at Scripps, had made breakthrough discoveries in the development of checkpoint inhibitors, which stimulate the immune system. And this resulted in drugs like Yervoy and Keytruda and Opdivo, really important medicines that have changed how cancer patients are treated. And it sparked this renaissance in interest in immunotherapies, and it led to them sharing the Nobel Prize in 2018. And so now we're entering this era of gene and cell therapy that I'm going to tell you about, where we're not just stimulating the immune system, but we can actually use genetic engineering to re-engineer it to do exactly what we want. So how does this work? How does the immune system recognize and eliminate tumor cells in the first place? Well, it uses this remarkable cell called the T cell, and Jamie mentioned this in the beginning. T cells are constantly patrolling every tissue of your body, your skin, your gut, even in the brain. And they're looking for signs of infection or cancer. And the way that they're able to distinguish a healthy cell from a potentially dangerous cell is with this here, is with the T cell receptor. These are remarkable. The T cell receptors it's almost like a combination. It's looking for the right key until it finds one where it activates the T cell. Theoretically, the T cell receptor can produce 10 to the 15th to 10 to the 20th different combinations. That's a quadrillion to 100 quintillion different combinations to protect you against a threat. Now, in practice, any one person probably has around 100 million different TCR clones at any one given time, but it's still a remarkable potential of these T cells. And what's amazing is that they're able to do this autonomously. So one cell can go around from cell to cell, and it can decide whether or not that cell should be eliminated or whether that cell should be saved as a normal or healthy cell. When it finds its target, when it finds a malignant cell or an infected cell, it does two things. The first is that it multiplies itself thousands of times over in order to overwhelm the threat. And the second thing that it does is it kills the cell that's infected or cancerous. So what's also very interesting about T cells is that after the threat is gone, the T cells then form a memory. And that memory of that antigenic threat can last for years. There's T cells from smallpox vaccines that have lasted for 50+ years. So this is a really interesting feature of the T cell, and it's why we chose the T cell as the basis for the therapy that I'm going to talk about now. So what's the problem? So T cells are really interesting, but cancers are really tricky. And they evolve ways to cloak themselves or to hide from the T cell or to get around or to exhaust the T cell or to suppress the T cell's activity so that they cannot be lysed by the T cell. This is where the genetic engineering comes in. We can rewire the T cell and force it to recognize the tumor. Chimeric antigen receptor T cells derive their name from a Greek mythological creature called the chimera. A chimera was a legendary fire-breathing hybrid that had the body of a lion, the head of a goat coming out of its back, and a tail that was a snake. That's kind of what we're building here with CAR T cells. CAR T cells are created by taking a patient's own T cells and creating a chimeric gene, which is comprised of this part of the T cell receptor, which allows the T cell to activate and to lyse tumor cells, and a part of an antibody that has a defined antigen specificity. And we define what that thing should target. We define what that antigen specificity should be. So in a way, we're kind of hijacking the T cell to do what we want. We deliver the gene into the T cell, into the genome, using a virus. And once that protein is expressed to the surface of the cell, now every time this T cell sees the antigen which we defined, it'll activate the T cell. And when it's in the body, it will serve as a mechanism to seek and destroy any antigen-positive cell wherever it is in the body. So this is how it looks in practice for a patient. A patient comes into the clinic. They have their white blood cells removed via a process called leukapheresis. We bring that into the lab. That's where the genetic engineering happens. Those cells are expanded in the lab. I'll show you a little bit more about this later. And then they're sent back to the patient. And before infusing into the patient receives a lymphodepleting chemotherapy to clear out the endogenous immune system and to make space for these incoming genetically engineered cells. The efficacy of these products is really remarkable. In patients that have failed multiple prior lines of therapy, tumors can be eliminated in just the first 30 days. So here's an example of a PET scan from a patient with diffuse large B-cell lymphoma. And you can see at baseline here, they have a lot of disease in the retroperitoneal lymph nodes. After just one dose of the CAR T cells, the disease is completely gone within 30 days. And again, this is a patient that's failed multiple prior lines of therapy. These results are so drastic. There are anecdotes from the radiologists that were reading these scans in early trials that thought this was the wrong scan. They thought this was a different patient because they'd never seen anything like this before. And this is because it's so much different than chemo and radiation. Instead of giving a dose of radiation where after you leave, you no longer have the radiation in you, or a chemo which you eventually will secrete, right? It has a half-life. CAR T cells do the opposite. They expand in the body into many, many, many-fold more than what you put in, and they overwhelm the tumor in order to eradicate it. The real watershed moment for the field was this case of Emily Whitehead. And you may have heard of Emily. She's been quite publicized. She's the poster child for CAR T cells. Emily was diagnosed with ALL leukemia at the age of 5. After 2 years of really aggressive chemo, her cancer stopped responding, and the oncologist estimated she only had weeks to live. She was out of options, but her parents were incredibly persistent. They found a phase 1 trial at the Children's Hospital in Philadelphia being run by Dr. Grupp and Dr. Carl June. And at the time, CAR T cells had never been tested in a pediatric patient. Emily was the first child to ever receive this type of therapy. And it's remarkable. Despite the fact that Emily had failed all the prior treatment options, she had a complete response to CAR T cells. And there's this story about Emily waking up from a medically induced coma on her 7th birthday with the care team at CHOP in Philadelphia singing happy birthday to her, and with her scans that showed she was cancer-free. And her father takes this picture every year to commemorate that moment. And I've talked to her dad, and the miracle here is the strength that he and her mother had to enroll her in a trial like this, being out of options and not knowing what was going to happen. But we're all really glad that they did, because what we learned from Emily has helped define the field of CAR T cells. Her case sparked global momentum for cell therapies in general, not just CAR T cell therapies, and it ultimately led to the approval of this product from Novartis called Kymriah in 2018. That was the first gene therapy approved by the FDA. Before Kymriah, only 10% of patients with relapsed refractory ALL leukemia survived 5 years. After Kymriah, it's almost 50% of patients. So these are not incremental changes. This is really paradigm-changing work that was done. But obviously, there are still challenges, and Jamie alluded to this. This therapy almost killed Emily. The reason she was in a medically induced coma is because she developed something called cytokine release syndrome, which she ultimately recovered from, but the toxicities can be really serious. And this is the fire-breathing part of the chimera. The toxicities typically have two manifestations. Cytokine release syndrome, or CRS, is the first, and that's typically accompanied by flu-like symptoms where a patient has a fever, they can occasionally have hypoxia, they can get very sick, but we can treat this with an anti-IL-6 therapy called tocilizumab, so we're blocking the cytokines. You may be familiar with CRS, or cytokine release syndrome, from COVID, where they use tocilizumab as well. This is similar. Instead of overactivation of the immune response to a virus, this is an overactivation of the immune system from CAR T cells that are expanding vigorously within the body and releasing cytokines into the body and making the patient sick. The other manifestation is more serious. This is neurotoxicity, or what we call ICANS. This leads a patient to lose fine motor skills, and it can eventually lead to cerebral edema and death. And one of the things that the patients lose, the first thing they lose the ability to do is they actually lose the ability to write. And so now in the clinic, what we do is every few hours, we have the patient write something down so that we can monitor their progress. So now we're 100 years on from William B. Coley injecting bacterial toxins into patients. We've got the sophistication to genetically engineer the immune system to go after the tumors that we want, yet we've still got this fire-breathing dragon that's creating the same toxicities that really William B. Coley saw over 100 years ago. So how do we solve this? And this is where our switchable CAR T cell platform comes in. We've designed this to harness the power of the genetic engineering that makes CAR T cells so effective, but to control the potential for immune-related adverse events. The concept is pretty simple. Rather than wiring the CAR T cell so that it's always on, we've installed this switch molecule such that the CAR T cell only works now when you have the switch. And the switch is made from an antibody, as I'll show you, and it's dosed separately from the CAR T cells. If there's an adverse event, we stop dosing the switch. The switch clears and the CAR T cells then turn off, because endogenously, these CAR T cells are off. They don't actually target anything. They only use the switch to target. In a way of thinking about this, it's restoring the pharmacology of traditional drugs to this gene therapy now. And so it's giving us the control that we typically had with conventional medicines. If we can successfully do this, there are advantages beyond safety. We believe that there are advantages that we can also get this to work in solid tumors, because to date, CAR T cells have only worked really in hematological malignancies, in blood cancers like leukemia and lymphoma. Ultimately, you can envision leveraging a platform like this in like a software and hardware-based approach, where our CAR T cells now become the hardware, and our switch molecule, again, based on an antibody that's dosed separately, becomes our software. And that software programs the CAR T cells where to go, what to target, how long and how hard to turn on, but most importantly, when to turn off. And we believe that this will allow a universal platform to be created to do this. Here's a molecular animation of what that looks like at the protein level. You see the switch molecule here, it slots in between the CAR-T cell and the target cell. And in doing so, it forms this immunological synapse. And when that synapse forms, you see these particles that get released from the CAR-T cell here. These are perforins and granzymes. And when they hit their target tumor cell, they cause the tumor cell to lyse open. This is exquisitely specific. So if the switch is not there, the CAR-T cells will not engage. So what does this look like for us experimentally in the lab? To do this, we had to design these proteins, these antibody-based switches, to fit into that immunological synapse sandwiched between the T cell and the target cell. And that distance is roughly 100 to 150 angstroms. So we had to do this with a great deal of precision. At that level of precision, doing this and predicting what it's going to look like is very difficult. And so we did this empirically by creating libraries of these antibody-based switches. And if you notice here, it doesn't look like a traditional antibody because we're just using the FAB portion of the antibody, which is just one arm of our antibody here. And we're targeting CD19 as a proof of concept. And the key here is you want to create the optimal immunological synapse. And if you create a synapse that's too long, you can imagine the CAR-T cell just doesn't get close enough to its target cancer cell in order to lyse it. And if you create one that's too short, sterically things don't work. You just can't fit everything into the immunological synapse. So it's a bit of a Goldilocks scenario to get this to work. But once we find the right switch, we can then use it like the software and run it like the program on the CAR-T cells. So this is looking now at CAR-T cells mixed with cancer cells in a dish under a microscope. The CAR-T cells here are in red and the cancer cells are in green. And you can see as soon as you add the switch, the CAR-T cells wrap around these green tumor cells. They cluster, they cause everything to cluster. And as time goes on, in just about 12 hours, all of those clusters turn to red because we've lysed away all of the tumor cells. The next step was to show that this worked in animal models, and this is how we run our animal models. We first inoculate a cancer cell into the animal. In this case, it's luciferized, so it glows under IVUS imaging. And this is a systemic blood cancer, so this is why it looks like it's systemically throughout the animal. We then give one dose and one dose only of our switchable CAR-T cells, followed by cycles of that antibody-based switch. And you can see without treatment, the tumor overruns the mouse within about 2 weeks. With treatment, these animals can go out, this is 80 days here, but they can go out to over 100 days. But we already knew this. We already knew CAR-T cells were incredibly efficacious. Our goal was to create something that could mitigate the toxicities. So I previously mentioned the key toxicity is cytokine release syndrome, which results from excessive cytokine production. Here we're showing that different doses of that antibody-based switch molecule targeting CD19 gives you a decreasing amount of cytokines. So we can actually accurately control the level of activation and the level of cytokines that are coming from these CAR-T cells, while at the same time getting the efficacy that we get from conventional CAR-T cells. We call it a switch, but that kind of makes you think of just on and off. This is actually more like a dimmer you can think about, or a volume knob that can tune or titrate the level of activity to a precise amount. One of the strengths of Calibr-Skaggs as a nonprofit is that we can collaborate with everyone. And so one of the first things we did is to collaborate with groups around the world to demonstrate that this doesn't just work in blood cancers, but it works in solid tumors like pancreatic cancer, prostate cancer, and breast cancer. And in every case here, we showed that we could completely eliminate the tumors in mice. So now we've got something in the lab that we know works. How do we get to patients? And this is where the Calibr-Skaggs infrastructure comes in. We built the teams necessary to translate these findings. We have the expertise internally to identify what is our lead drug candidate from this. We've got the teams that can run the toxicology models to understand whether it's going to be safe. We've got the teams that do the manufacturing of the drug product, and in this case, have learned how to do cell manufacturing from scratch. And then we've got our clinical regulatory teams that understand how to move this into patients and get the FDA to allow us to move forward. And that's what we did here for our first trial of switchable CAR-T cells targeting the CD19 antigen for patients with leukemia and lymphoma. We partnered with AbbVie, provided us with the funding in order to move this forward. And in return, AbbVie took an option to the program to potentially license it after we de-risk it. So this is where the synergy comes in with major pharma groups. We started this trial at the height of the pandemic. I think I described how the front end of this works for a patient already. The patient comes in, they're screened, they have the leukapheresis, the white blood cells are sent to the lab where they're manufactured, they're brought back to the patient, and after lymphodepleting chemotherapy, they're then given cycles of that antibody-based switch molecule. Now, the cells here are now called CLBR001, and the switch is called SWI019. This is how it looks like in our manufacturing facility, and this is the equipment we use to do this type of genetic engineering. When we receive the leukapheresis, we first separate the T cells into two types. They come in CD4s and CD8s. CD8s are the cells that are lysing. CD4s are the helper cells that are helping them lyse. We then transduce them with this lentiviral vector that contains the gene, which contains the CAR that will transduce them into a switchable CAR-T cell. And then this is how we grow them. We grow them in these 1-liter G-Rex bioreactor flasks, and we grow them up to billions and billions of cells. And this is actually the shortest period of time that we have. This is only 8 days, so it only takes about a week to do this. And then we spend the next 2 to 3 weeks testing them to make sure that they're safe, that they're not contaminated, and that they can be returned to this patient as a high-quality product. This is a highly personalized medicine. So this is an autologous CAR-T cell product. We do this for every patient on the trial. No two patients' products are the same here because it's the patient cells that are actually the drug here. This is what the responses look like in our patients. So our first patient, I'm happy to say, was enrolled right here in San Diego at the UCSD Moores Cancer Center. And I think we take a lot of pride in the science that's done, not only here at Scripps, but here in San Diego on the Mesa collaboratively. This patient is included in this data here. So each one of these lines is a patient's tumor burden that's shown by assessing lesion sizes by PET scan over time. And the gray boxes here are our cycles of our antibody switch. And we're giving the CAR-T cells at the beginning. You can see first that after the first 2 cycles, a lot of the patients have really significantly come down in tumor burden. In fact, we're getting complete responses just after the first 2 cycles. But what's more important is that as we continue to dose over time, we're continuing to get deeper and deeper and deeper responses from these patients. Here's a case study of one of those patients. This is a 71-year-old man. He failed 6 prior lines of therapy. He was treated with the switchable CAR-T cell. At baseline, he had a large mesenteric lymph node mass. And you can see after 3 months, it's almost gone. After 6 months, that's just scar tissue that's left. He's had a complete response here. And what's wonderful about this patient is he did not have any cytokine release syndrome or any neurotoxicity. So he's able to enjoy the response without the toxicity. Overall, our responses are similar to what we see from the conventional products that I showed you previously. We have about 75% or so in this small trial that responded, and about half of those patients had complete responses of their disease. And while we're not completely eliminating all CRS and neurotoxicity like I showed you with that last patient, the important thing is here that patients that do experience those types of toxicities have a very short duration of the toxicity. So for CRS here, in the pivotal trials for the commercial products, these patients have CRS for about a week, and even more concerningly, it's about 2 weeks of neurotoxicity. This is the big concern for patients. Nobody wants to have altered mental state and confusion and the inability to write for 2 weeks. In our trial, we got this down to just 1 day. And that's because we can stop dosing that switch at the first sign of toxicity. The other thing that we can do is we don't have to stop dosing it. We can also decrease the dose of it. And you might say, okay, well, Travis, one of the reasons why you're not getting a lot of toxicity is because your CAR T cells are weak. They don't really expand. And so you just have less of them, and less of them means not as many adverse events. Paradoxically, we have more CAR T cells. We have 10 times more CAR T cells in the patient. They've expanded 10 times more than a conventional product. And this is because when we're dosing with the switch, we're giving this cycle, this pulsatile on, off to the CAR T cells. And that's actually helping them to expand and preventing their exhaustion. So we're able to get more therapy in with fewer incidents of prolonged adverse events. Here's another case study to show you what this looks like for a patient. This is a 70-year-old female. They failed 7 prior lines of therapy. There's a lot of data on this page, but you can see here in the cycle 1, cycle 2, cycle 3 of the switch that at the end of cycle 1, they began to develop a low-grade CRS. Grade 1 CRS is basically just a fever. The switch was stopped at the same time that cytokines were peaking here in blue. And what's important is that the CAR T cells didn't go away. So after we stopped dosing, the CAR T cells actually remain. And in this case, they continued to expand, but they were not producing an excessive amount of cytokines anymore. So this patient's CRS ended, they were released, they went home, they came back in for their second cycle. And this time they again had a low level of CRS, and this time a low level of neurotoxicity. But instead of stopping this time, what we did was we reduced the dose of the switch in half. So we went from 10 microgram per kg to 5. And now this patient was able to go on to receive additional cycles of the switch and enjoy a complete response to the therapy. At the time that we obtained this data, this was the first example of controlling a genetically engineered cell in someone's body where you could turn it on, off, on again. No one had ever done this before. This is really interesting data. This is looking at individual patient CAR T cells over time. So this is in peripheral blood. We're taking a sample of their blood, and we're saying, how many CAR T cells do you have in your peripheral blood over time? And you can see in this case here shown in orange that the CAR T cells, they come down to this relatively low level. And then after almost a year, they had a relapse. And we were able to come back in with another dose of that switch molecule without giving them any more CAR T cells. And we saw CAR T cells in peripheral blood came roaring back. So that's indicating these cells, after therapy is done, are actually remaining in the patient. They're persisting. They're engrafted into the patient, almost like a memory T cell. They are memory T cells. And it indicates that these patients now have a long-lived, genetically engineered component of their immune system that can be recalled in the future in the case of relapse. And the vision is that one day we might have a library of these switches that if the tumor changes, we could go after a different antigen and hit a different antigen in order to eliminate whatever changes have happened to the tumor. And we only need to do that by giving the software because the patient already has the hardware. That approach is going to be critical to get this to work in solid tumors. I mentioned it works phenomenally well in blood cancers, but it does not work very well in solid tumors. And the problem is multifactorial. First, solid tumors have an immunosuppressive tumor microenvironment. So it makes it very difficult for the CAR T cells to activate and to lyse tumor cells in these environments. The tumor cells, they're creating exhaustion of these CAR T cells. The second is that solid tumors commonly share antigens with vital or other healthy tissues. And so the toxicities are really severe if you try to target them. And the third is that solid tumors are commonly very, very heterogeneous. And so just targeting one antigen doesn't really work for you. But if you think about the advantages that we've now developed for switchable CAR T cells, we can expand more CAR T cells in a pulsatile fashion, which is avoiding the exhaustion. So we might be able to overcome the immunosuppressive microenvironment. We can turn them off in the case of an adverse event, or we can tune or titrate their level of activity. So we might be able to tune them to a level where they target the tumor cells and not the healthy cells. And then we're able to potentially load multiple software programs in via multiple switches to tackle the heterogeneity. And so based on this data, we launched a clinical trial in breast cancer in collaboration with AbbVie. This trial has just started. And what's exciting about this trial is not only will this be the first time we've treated patients with a switchable CAR-T cell in breast cancer, but we're targeting an antigen here which has never been targeted before by an immunotherapy, which is the prolactin receptor. So this is a first for two things in this trial. One of the last things I'm going to tell you about is how we can get this to work for autoimmune diseases. And you might be saying, well, it works for cancer. This seems the opposite in autoimmune, and it is. In autoimmune, T cells are not missing cells to target. They're targeting too many cells. They're causing inflammation in healthy tissues for these native T cells. So how do we translate this to autoimmune? Well, we use the CAR T cells against the pathogenic cell, which in this case is the B cell. The B cell is what's producing these autoantibodies that's leading to the patient's illness. And early data has already shown that if you can completely eliminate the B cells, you create this global immune reset. And that global immune reset actually puts patients, especially with lupus, into deep remissions of their autoimmune diseases. And you can see some of the first data here. You don't need to be an expert in reading these scores to see their disease scores come down by 3 months, and by 6 months, the patient has no disease. These patients are now out years, lupus patients, SLE, without the need to take any more immunosuppressive therapies to control their disease. So this is really, really quite amazing. So what can we do with the switch? Well, with the switch, because we have this advantage of the ability to expand all of these cells to great numbers, and because we're pulsing them on and off, we can actually control the safety, first of all, but we believe we can do this without that lymphodepleting chemotherapy that I mentioned before. Remember that all cancer patients receive a lymphodepleting chemotherapy in order to make space in their endogenous immune system for those adoptively transferred engineered cells. In autoimmune patients, the risk-benefit profile is much different. These patients are typically, they don't have terminal disease. They're typically much younger, and the impact on quality of life is typically much higher, for instance, loss of fertility. And so if we can eliminate the need to use lymphodepleting chemotherapy in these patients, then we really might be changing the game for patients with autoimmune diseases. So we've opened this trial. It's enrolling lupus, myositis, systemic sclerosis, and I'm excited to say we just opened it for RA patients as well, because it's a really big unmet need. The last thing I'm going to tell you about is kind of the elephant in the room. This is an incredibly time-consuming, labor-intensive, and expensive process to make CAR-T cells. And so it's restricted right now to doing it in highly specialized centers. Not everyone has access to a highly specialized center. So how do we get this to the community hospitals where we can really democratize it and get it to work with multiple diseases and multiple people? The answer is to eliminate the ex vivo part of this manufacturing, eliminate this manufacturing facility, instead manufacture the cells right in the patient. So instead of taking the virus and transducing the cells in the dish, we're taking the virus now and we're going to try to deliver it directly into the patient. So it's just a single dose of this. There's no waiting for the manufacturing to be done. There's no cost involved in manufacturing. You can do this what's called in situ. So it takes an extraordinary team to do something like this. It's driven in large part by Pete and his infectious enthusiasm for doing really hard things that most other people would simply run from. Infectious is the only word I can think of, because I don't know how he convinced us to make, a cell manufacturing process from scratch. But we're very fortunate to be in an environment like Scripps with a lot of talented scientists who are living and breathing these challenges of discovery and drug development on a daily basis. There's really no other place like this. I should point out that many of the folks on this slide came to Calibr as postdocs. And Jamie mentioned the strong training mission that we have here at Scripps. We do at Calibr as well. We've trained these very talented folks in drug discovery, and they've gone off, and many of them actually come back, and they're running their own groups now at Calibr, which is something we're very proud of. Obviously, we would be nowhere without the dedication and the bravery of the patients who have enrolled in this trial, the caretakers, the physicians, and their families. Cancer is something is very personal. Jamie opened with that. It's very personal to many of us. My journey with cancer became very, very personal to me when I lost my father last year to colon cancer. And at the same time, I learned that he carried a heritable mutation, which increases my potential risk for cancer. and my children's potential risk for cancer. And so I feel the urgency to get these solutions in a very personal way. And I can't tell you how appreciative I am to work with a team here that has that similar sense of urgency to solve these challenges. Happy to take any questions. I think we're going to sit down with Jamie, and if there's time, we'll take some questions. So thank you.
Great. Okay, terrific, Travis. Thanks so much. So there's, if you have a question, stick your hand up. I'm going to kind of open with one that struck me. So you described a lot of the people in your first cohort of patients, and they all had failed therapy multiple times. So in a way, to enroll in this trial, you had to be sort of last resort. And so I'm kind of wondering, if people who have never been exposed to other immunotherapy or chemotherapy, that they're much earlier in the whole treatment cycle, will they enjoy any advantage for doing this switchable CAR-T sooner than having it kind of be at the end of a journey?
Yeah, that's a great question. And it's clear with all immunotherapies, not just CAR-T, that the sooner you receive immunotherapy as a patient with cancer, the better it seems to work. Because after chemo, the immune system seems to wane. And after the tumor becomes larger, it seems to impress the immune system in a negative way. And so earlier CAR-T cell therapy is absolutely more efficacious. And if we can make it potentially safer with the switch, we can potentially move it to earlier and earlier lines.
But so is there some kind of regulatory hurdle because you've got standard of care or new experimental treatment?
Yes.
And so how does a patient navigate that?
Yes.
Yeah,
that's always a challenge, and it's a good challenge to have, right? As therapies get better and better, it becomes harder and harder to get your therapy approved or your therapy moved up in the lines of therapy because the bar is set so much higher. And so the bar has been elevated by conventional CAR-T cell products, and now we will have to show that we have an advantage, whether that's efficacy or we have the same efficacy, but we potentially have a differentiation in safety.
Okay, must be intrepid questions from the audience. There's one. We got it.
Do you have any indication yet of how effective this is for a mantle lymphoma, the mantle tumors?
Can you say that again? Did you say?
There's a lymphoma called a mantle tumor, and I wondered if you've...
Yes, mantle cell lymphoma. Yes, in fact, mantle cell lymphoma was, there were patients that I showed you today that had MCL, mantle cell lymphoma. Yes. Yes.
There's a question over on the wall. Yeah. Yes.
Do you get long-term depletion of B cells with this? It seems like you wouldn't, right?
That's a great question. So it's not clear how long B cells need to be depleted in order to get this effect. With the conventional products with blood cancers, one of the problems that they had were patients were depleted of B cells for a long time. That was with the cancer trials. With these autoimmune trials, for whatever reason, the B cells seem to come back. But they're no longer pathogenic. And that's why it's called an immune reset, because even though the B cells are coming back, the disease doesn't.
And that's with conventional?
That's with conventional CAR-T cells, that's right. And so these results are relatively new. There haven't been a ton of patients that have been followed for a long period of time. So the field is still trying to understand why that is.
Yeah.
CD19 CAR-T cells around.
In some cases, yes, yes.
Down here in front.
Hi. I would imagine this might be case specific, but I'm curious how you determine the time after administering these SCAR-Ts to then administer that antibody switch, or if it makes a difference after a period of time has passed.
Yeah, I'll repeat that question. So the question was, how long after delivery of the CAR-T cells do we wait before delivering that antibody? And that's a very complicated question. In the first trial, one of the challenges we faced from a regulatory perspective was neither one of those pieces, neither the CAR-T cells nor the switch, had ever been in a human before. And it's very rare to do a trial in which you're dosing two things that have never been treated before. Usually if you're combining two things, there's at least safety on one of them. And so one of the ways that we came up with to do that was to actually vary that time between delivery of the CAR T cells and delivery of the switch. Ideally, we want to give them right away, right? Because we don't see a need to wait therapeutically. But for a safety perspective, we did wait in our first patients to make sure that those CAR T cells did exactly what we wanted them to do, which was nothing in the first couple of days. Great question.
So Travis, I'm going to aggregate. I'm getting questions from the virtual audience here. So, but I'm going to. There's a lot of them, but I'm going to kind of aggregate it. Now you touched on the autoimmune application, but could you say a little bit more about where you see the specific, the best shot for treating specific conditions, and and what have you tried? Just kind of recap on that.
Yes, by far. They work the best so far in lupus. Mm-hmm. Lupus patients have enjoyed the best benefit. It's not approved for lupus yet, but in the clinical trials, the lupus patients have been by far the best responders. In other indications, systemic sclerosis, myositis, they do seem to work, but we don't know if CD19 is the perfect antigen for that. It may require other antigens. And there's other companies that are going after really rare diseases, like stiff person syndrome, which Celine Dion has. And actually, I think the company that's doing it is going after it because Celine Dion has it. But there are a number of B-cell driven diseases that are quite rare that we believe can be helped by this therapy. But right now, lupus is responding the best, myositis, systemic sclerosis, RA, some other diseases are also responding.
So I was really interested in the data you showed toward the end, where these CAR T cells are persisting for a long time. So what controls that? I mean, they started out as your normal cells, you reprogram them, but what controls their level of replication, and when do they decide to go to sleep? And
this is a great. I mean, I think it's a good question because the way we think this works is similar to if you got sick one day with, let's say, a new virus that you hadn't been experienced at before, and let's say it's the first time you got, let's say, COVID or something, right? Your body's going to respond. Hopefully it will clear that, but then those cells will remain within the body waiting until the next time you get exposed to that virus. And the next time you get exposed to the virus, hopefully you won't get as sick anymore. So the cells, our CAR T cells, are doing, we hope, the same thing that those typical memory T cells do, which is they go to the secondary, your lymph nodes, or your spleen. That's where they kind of hang out until they see that antigen and they say, aha, they wake up.
So that, you touched on this, that brings me to this other kind of question. So you put these cells in and they do nothing until you give the switch. And you alluded to this, but you can use a different switch, right? So you can go in, if you have one kind of cancer and you treat it, if you get another kind of cancer, you already have the CAR Ts, and you can now just give a different switch without reintroducing the CAR Ts.
That's right. That's the vision.
Has that ever been done in, I mean, are you?
We've done that in animal models. We have not yet done that in patients. And we're getting there. We have the library of tools to do it now in the lab, and the next step would be to progress it in order to do exactly that.
So you're just primed for anti-cancer therapy once you get this.
Yes, and I think the first place, like I said, to do that will be solid tumors. Because in solid tumors, you typically have a lot of heterogeneity, where if you eliminate one antigen-positive cell, there's another one that's still there.
Okay, we have a question over here.
Fantastic, very engaging talk. Thank you very much for that. Curious whether you know what is the frequency of the maximum, to achieve the maximum efficacy in these patients. Like how frequently do you have to dose the switch, and does it degrade, completely excretes between those doses, or does it have to be like at a sustained level to get the maximal efficacy?
Yeah, this is something I didn't get into, but is an important question. We have found, so right now, if you noticed in the cycles, we're dosing for 1 week, followed by 3 weeks of a dosing holiday. So it's a 28-day cycle during which time the patient is receiving 1 week of therapy and 3 weeks of nothing. We found, and we published this in our preclinical models, that 3 weeks of nothing is more important than the 1 week of dosing. It's very counterintuitive. The rest phase for the CAR T cell is critical. And that's what you don't get from conventional CAR T cells. Although they work very, very well, those cells are never resting because they're hardwired to be on. And it was interesting that when we first developed this, and we had not yet gotten to the phase where we got proof of concept, I would go and I'd give this talk at conferences, and people would say to me, but when you stop dosing that switch, your CAR T cells, they're going to go away. You're going to lose them. Because the conventional wisdom at the time was you have to have constant stimulation to keep those T cells in the body. I mean, this is what you were getting at as well. And we said, I don't think so. I think when you get sick or when you get a vaccine and you give this high load antigenic stimulation to the T cells and then you rest them, that's the physiological way a T cell likes to respond. And so to your question, what's the frequency? Actually, for the T cell, the longer the rest period, the better.
Okay, I think we're coming up on the hour, and so we can continue this discussion for the in-person audience out at the reception. There's a science in action table. We've got staff from communications and philanthropy that's out there to have a discussion with you. And the last thing I need to call your attention to is the next front row, which is going to be Ben Cravat, one of our stars working on finding new drug targets. And that's on September 17th. And so if you come here on September 17th, as is my custom, you will find me in the front row. Thank you.